So the important takeaway here is that viewing red light and near-infrared light at a distance at which it is safe for just a couple of minutes each day allowed a reversal of the aging process of these neurons, which some people have heard me say before, and I'll just say it again. The retina, including your photoreceptors, are not just connected to your brain. They're not just near your brain. They are actual central nervous system tissue. They are the only two pieces of your brain, meaning your neural retinas are the only two pieces of your brain that reside outside your skull, or at least outside the cranial vault. So here we're seeing a reversal of the aging process in neurons by shining red light on those neurons. Now, of course, the Jeffrey Lab is primarily interested in vision, and humans are most dependent on vision as a sense to navigate the world and survive. So this is really wonderful. Here we're looking at a therapy that can reverse age-related vision loss, at least in some individuals. But as you can imagine, the study was also done on these cells because they reside outside the skull, and you can shine light directly on them, right? I'm sure that there are many people out there who are interested in how they can improve the function, say, of the neurons in their brain responsible for memory. And in a few minutes, I'll describe the non-invasive applications of light to try and restore the function of those cells as well. So a little bit more about the studies from the Jeffrey Lab. One of the things that they observed was a reduction in so-called drusen, D-R-U-S-E-N. Drusen are little fatty deposits, little cholesterol deposits that accumulate in the eye as we age. We've all heard about cholesterol within our veins and arteries and how that can clog our veins and arteries, and how, of course, clogging of veins and arteries is not a good thing. Well, our neural retina, being so metabolically active, requires a lot of blood flow. It's heavily vascularized. And drusen are a special form of cholesterol that accumulate in the eye. As it turns out, these red light and near-infrared light therapies explored by the Jeffrey Lab were able to actually reduce or reverse some of the accumulation of drusen. And so in addition to reducing reactive oxygen species, the idea in mind now is that red light may actually reduce cholesterol deposits and reactive oxygen species in order to improve neuronal function.